Welcome back to another exciting episode of Teach Me in 10. This is the video series that's brought to you by Technology Networks, where we sit down with experts who share their knowledge with you in under 10 minutes, which gives you nice bite-sized, insightful content. And in today's episode, I am delighted to have Marrit Putker as our guest. She leads the In Vitro Research and Development team at Crown Bioscience in the Netherlands. And as the episode title might already suggest to you, we're diving deep into Crown Bioscience's cutting-edge drug screening platform. This new assay-ready Organoid platform enables high-throughput drug screening and large panel screens in record time. Before we go any further, I am excited to say that this episode is kindly brought to you by Crown Bioscience. Crown Bioscience provides life science tools for translational research and preclinical drug discovery that enable a wide variety of in vitro and in vivo applications. To learn more about Crown Biosciences, you can click the link that we've left in the description. But without further ado, get ready to enjoy this incredible Teach Me in 10 in full. Hi Marrit, how are you doing?
Hi, Lucy. Very well, thanks. How are you doing?
Wonderful. And as we only have 10 minutes on the clock, we best get started. So my first question has to be around organoids. They're pretty new players in the field of preclinical drug testing, but I'm really interested in what your opinions are on the major advantages of using them over other available models.
Sure. So, well, organoids that we're using, they're actually established from adult stem cells, right? So they have the advantage of being cultured in a very native state. So we're kind of keeping the tumor tissue as well in a very relative primary state. So when you compare this to, for example, cell lines, the conventional tissues that we're using in the drug screening platforms in vitro, they've actually been adapted to growing in 2D, and they've been adapted to growing on plastic. So there, in particularly, the differences are very large. So organoids remain very similar to the original tissue of origin, both genetically, but also when we look at the morphology or when we look at gene expression levels. And in particular, also importantly for us, in their drug responses. So they really remain very predictability with regards to their tumor tissue is very, very high. And then if you look back into other models more preclinically and you go wider, of course, you can also look at in vivo models. And there, the big advantage for organoids would be the scalability. So we, of course, have the ability to grow them in very large amounts. So we can go high throughput. We can test many more models at the same time, which is also something we allude to later in today's, probably in today's chats. And then we can also do combination screens, which makes a big advantage over other models.
That makes a lot of sense. So then, how robust and reproducible are the data that you produce with your organoid drug screening services?
So overall, the robustness actually is very high. So it's nicely illustrated with the performance of our experiments. So when we look at our standard drug assays, where we do the cell-target glow assays, for example, it's very easy to measure, but also with our high-content imaging platform, we reach actually assay performance with very high Z factors, often averaging 0.7. We reach very low control variations, and we're including also some standard of cares, which we try to repeatedly include so that we can compare them to previous experiments. We've done this a bit more elaborate in the past year, where we've compared the drug responses over, for example, 10 passages. So really taking the same culture, every passage again and again, test the same drug response. And we would find very similar IC50 data when testing standard of care drug testing. So that I think nicely illustrates that these models in general are very robust. Of course, there can be differences there. We've been optimizing our procedures even further as well by actually allowing repeated measures from the same batch. So we're batching up the organoids larger, and we can do repeated measurements. So thereby we can even more increase the reproducibility of our assays.
Nice. And then from that, I'm really left wondering, what are the limitations of the organoid technology for preclinical drug screening?
Yeah, there's always, of course, limitations. I would say in this case, since we're looking at stem cells from the adult stem cells, the technology is really limited to epithelial tissues, which makes that we can only measure or at least test in these drug assays with the organoids only epithelial tumors. So that means that we cannot, for example, test hematological cancers or sarcomas. So we're really looking only at carcinomas in this sense. And also, you can view this as a limitation or as an advantage or as an opportunity. We're only growing the epithelial cells, so the TME is not available in the cultures that we're growing, meaning that we can really specifically look at epithelial cells, and we can then add in and reconstitute TMEs later. So it's a limitation, but you can also see you can selectively add the cell types that you're interested in. So this can also be an opportunity.
Definitely. And then I'm interested in the key differences between PDOs and PDXOs when it comes to drug screening. Are there any specific advantages or disadvantages to each?
So yeah, let's just first briefly explain. I think a PDO stands for patient-derived organoid, where a PDXO stands for patient-derived derived organoids. So those are organoids which we derive from our PDX models. So we take the mouse, take a human tumor, and then grow it into a PDXO. So in the basis, these models are similar. They're both derived from patient tissues. One has been grown in mouse before entering organoid stage. Both actually very nicely predict the response of the tumor. The genetics are very similar to the original tumor. So in that sense, those models are very similar. In practice, later onwards, there is a difference. So for example, for the PDXO models, since they have been derived from our PDX models, this also means that we have for all of these models an in vivo model for follow-up. So you could actually do a big test multiple PDXOs, and then say, okay, I want to test these 5 models. I want to do further testing in vivo. And that's a guarantee for the PDXO models. For the PDO models that we have, we are also building the PDO-X biobank, meaning that we are making xenografts from our PDO models. So we do have for many of our PDOs also in vivo follow-up, but not for all. The other way around for the PDOs, because they are directly derived from the patient, we can also, for some of those models, we've been able to also harvest the normal tissue, so the adjacent tissue. And that's one of the unique features of organoids, because we're growing those adult stem cells. We can also grow them from non-tumorous cells. And this is a very unique feature of the organoids in the preclinical space, where we can really compare normal tissue versus the tumor tissue. And that's really specific, because those normal cells will not grow out in a PDX model in the mouse, because they will just die. They're not cancer, so they won't grow in a normal tissue. But in our in vitro setup, they do. So that really gives a nice advantage of those PDOs.
Absolutely. And then finally, as we're almost out of time, what, in your opinion, are the major opportunities that these novel large-scale screening methods bring?
Yeah, so indeed, so we haven't really talked actually about the large scale. So I just mentioned we have been able to bank up our organoids larger, so we can actually make those batches and we can repeatedly do those assays. And this also allows us to indeed increase our throughput. So we can actually test up to, for example, at the moment, 50 models at the same time. So we can provide those data within 6 weeks after starting in a project. And this allows to do many organoids testing at the same time, which then allows biomarker analysis. So you're testing one compound against all 50 models, and you can say, okay, well, this model responded, this model didn't. Which biomarkers come with this response? You can think of mechanism of action in a similar way. But you could also think about drug repurposing, for example. We have multiple indications in our panel that we are introducing. So when you're testing your drugs over those different indications, you can maybe find patterns where there's indications which we didn't expect yet, that do actually respond. So you can either do that for drug repurposing or indication selections for INDs. As I just alluded, for the PDXO models, we actually have the in vivo models. So you could do, for example, you can use these screenings as well to select your in vivo models that you want to run in follow-up. And I think, yeah, the larger, of course, the panel gets, the bigger the impact becomes. But if we can run those 50 models or up to 100 maybe later, we can actually start thinking about running preclinical clinical trials, right? So actually already stratifying the patients based on those biomarkers upfront, and then allowing clients to already stratify their patients for clinical trials before they actually start a clinical trial. And thereby, well, testing the drugs on the right patients, tremendously improving the drug development pipeline.
Wonderful. Well, thanks so much, Marrit. It's been an absolute pleasure speaking with you today.
Thank you very much. My pleasure.
Thank you so much for joining us today, and I look forward to seeing you again very soon.